Yes. Thank you again for that introduction. I just wanted to start off with some quick background for my project. So as I'm sure everyone here knows, obesity is often measured with a BMI of 30 and above and is a major concern and projected to increase dramatically. It's associated with metabolic disorder, which is correlated with other effects such as increased cardiovascular disease, hypertension, stroke, and type 2 diabetes. Factors impacting obesity include diet, exercise, genetics, and the microbiome. So if we focus in on diet, dietary fiber has been shown to improve metabolic health and protect against obesity through still unknown mechanisms. So fiber is specifically an edible carbohydrate polymer with 3 or more monomeric units that are resistant to endogenous digestive enzymes and therefore are not hydrolyzed or absorbed in the intestine. Fiber can be characterized into two groups based off of their solubility, either insoluble fiber, which works to build fecal material and aid in gut motility, or soluble fibers, which are ingested and then can be metabolized by the gut microbiome. They have been shown to lead to increased gut microbial diversity and lead to many beneficial effects, including reducing the risk of gastrointestinal diseases. We're going to focus in on the role of soluble fibers for the purposes of my talk today. So additionally, I wanted to cover the gut microbiome, which is a large player in metabolic regulation. Many factors are known to influence the gut microbiome, which would then reduce diversity and cause dysregulation of the microbiome to lead to increased metabolic dysfunction. The gut microbiome influences host factors through interactions with dietary nutrients, such as fermentable fiber, which produces short-chain fatty acids. So these encompass any fatty acid with a carbon chain of less than 6, but the vast majority are composed of C2, 3, and 4, acetate, propionate, and butyrate. These can go on to act as major energy sources for the body. Short-chain fatty acids play a variety of roles in the regulation of metabolic homeostasis by providing energy for various organs, regulating glycemic control, and mediating immune responses. Importantly, they also act as ligands for G-protein coupled receptors, and it is thought that this signaling mediates most of their functions. The two main receptors I will be discussing are FFAR2 and 3, or free fatty acid receptor 2 and 3. As mentioned before, both receptors are GPCRs and they belong to the free fatty acid receptor family. They can sense and mediate the action of short-chain fatty acids throughout the body. These receptors are expressed globally, but also have tissue-specific expression. Both receptors are highly similar and have 43% amino acid sequence identity, but they differ in their expression profile, short-chain fatty acid binding affinities, and downstream signaling pathways. Additionally, FFA2 has an affinity for the shorter short-chain fatty acids, such as acetate and propionate, while F3 has higher affinity for the longer short-chain fatty acids, butyrate and propionate. FFA2 is associated with G-alpha-Q stimulatory signaling, while FFA3 is associated with G-alpha-I inhibitory signaling. Both of these receptors have recently emerged as therapeutic targets based on their ability to mediate effects of short-chain fatty acids, including metabolic regulation. So for the purposes of today, I'm interested in the whole body role of short-chain fatty acid sensing mechanisms and the complex interplay between the gut microbiome, short-chain fatty acids, and free fatty acid receptor 2 and 3. So kind of in summary, fermentable fiber can interact with the gut microbiome to produce short-chain fatty acids. These are globally recognized by FFA2 and 3, and overall this process has been seen to protect against metabolic dysfunction. So to start off with some preliminary data from our lab, we had previously conducted an 8-week dietary study which tested the effects of a control normal chow, which we see here in pink, along with a high-fat, high-sugar Western diet in yellow, and the same Western diet supplemented with 3 soluble fibers: guar gum, fructooligosaccharides, and pectin. So first, plasma short-chain fatty acid concentrations are measured. We have acetate here on the left, propionate in the middle, and butyrate on the right. So when the mice are fed a Western diet, we saw a significant decrease in short-chain fatty acids relative to the chow control. All 3 of the Western diet fiber supplemented groups saw a significant increase in short-chain fatty acids, again, relative to the Western diet this time. So when tracking body weight over the 8 weeks, we observed increased body weight on the Western diet, and this weight gain was reduced when the mice are fed fiber supplemented diets. Similarly, we saw improved ad lib insulin levels after supplementation with fiber here. So therefore, we established here that fiber plays a role in the protection against Western diet. And now we want to go in and investigate more of the mechanisms. From here on out, studies will be using FOS, as the mice seem to tolerate this fiber supplement the best. Another previous study in the lab followed novel FFA2, FFA3 double knockout mice for 10 weeks on a Western diet with or without fiber supplementation. Here we have wild-type mice on a Western diet alongside their fiber supplemented matches. Wild type again. We also have double knockouts here and here on the same 2 diets. After 6 to 10 weeks, about here, we see the desired protective effect of fiber in wild type mice. But interestingly, we do not see this in the double knockout mice here and again here. So this is suggesting the role of FFA2 and FFA3 in the fiber-mediated protection against Western diet. This is what I want to further understand the mechanism for in my project. So we know that receptors can impact energy expenditure, insulin and incretin hormone signaling, and these factors may be impacting protection against metabolic dysfunction. To do this, I have a 2-pronged approach. Aim 1 is focusing on the role of the receptors in metabolic dysfunction, while aim 2 is focusing in on the impact of fiber on the microbial diversity as a factor for metabolic protection, potentially even bypassing the receptors. So first, I want to focus in on the receptors. Based off of our previous study, we conducted a more in-depth study. We followed the same double knockout mice alongside a matched wild-type control for a 25-week-long analysis, increasing the time point to study the long-term dietary effects. These mice were raised on a normal chow for 10 weeks before being put on 2 challenge diets, either a Western diet or a Western diet supplemented with 20% FOS. Mouse weights were tracked weekly. We also did body composition analysis before the diet starts and at the end of the study. We conducted food intake at the middle of the study and metabolic analysis at the end of the study. Additionally, we studied insulin sensitivity and secretion at the end of the study with insulin tolerance and glucose tolerance tests. So here, what we were doing is we were thinking that the wild-type mice on fiber supplemented diets will be protected against metabolic dysfunction from Western diet, while the double knockout models would not see this effect. And we were trying to investigate the mechanism, thinking it's due to the action of FFA2 and FFA3 on either food intake or energy expenditure, or both. Oh. There we go. So here we can see the weight data through the study. We have here wild type and double knockout mice on Western diet, and wild type and double knockout mice on fiber supplemented diets. The left plot shows the overall change in mouse weights from their baseline alongside the weekly weight values on the right. We are seeing that the mice on the Western diet gain the most weight here, and again here. Only the wild type mice are significantly protected from the weight gain, while the double knockout mice on fiber still gain weight. For we have here replicated the weight phenotype seen previously and confirmed what we saw in the shorter 10-week preliminary study. We are seeing this echoed in the change in fat mass from the baseline. Again, wild type group on fiber here is protected from the increase of fat mass seen by the Western diet groups, alongside the double knockout group on fiber. This is confirming that the body weight trends are due to an increased adipose phenotype. We next looked at ad lib insulin at the endpoint of the study. We're seeing that it is decreased in both fiber groups, but the double knockout group is trending higher. We're not seeing much change in ad lib glucose values. Similarly, we see that fasting insulin is again is lower in wild type mice on fiber, but trending higher with the double knockout mice on fiber. We're not seeing much of a difference between the two fiber groups in fasting glucose, however. Next, I looked at insulin tolerance tests at the end of the study, showing that we see here that the wild type mice on fiber are the only group that show insulin sensitivity. The other three groups are more resistant. Similarly, the GTT data shows that the wild type mice on fiber, here again in brown, show the lowest insulin response along with the lowest insulin response here, along with the fastest glucose clearance compared to the other three groups. We're seeing this echoed in the area under the curve data. So with the food intake studies, we are seeing not much of a difference. There might be a slight higher trend for the fiber groups, but this needs more study. So moving on to my metabolic data, we have here average energy expenditure across either the day, the full day, or the light or the dark phases. We're seeing, interestingly, increased energy expenditure in the double knockout mice on fiber. I did an ANCOVA regression analysis, which showed that all four groups were falling on separate regression lines, indicating that the group effect here is not due to differences in body mass, but rather something else. This is again echoed in the hourly averages across the same 48-hour period, with the double knockout mice on fiber with the highest energy expenditures. Interestingly, the double knockout mice on Western diet, though not significant, are trending higher. Therefore, it could be an inherent effect of the mouse model itself. I'm planning on doing more baseline testing in my next cohort to see this further. This data is supported by the increased oxygen production and carbon dioxide consumption in the double knockout mice here. Oxygen consumption is on the left and CO2 production is on the right. However, we're not seeing much of a change in the respiratory exchange ratio, or the ratio between the VCO2 and O2. Generally, VCO2 and RER is between 0.7 and 1.0, where 0.7 is burning fat and 1 is burning carbs. Since they're all about the same, near 7.5 or 8, it looks like they're burning both with no differences between the models. The higher VCO2 and VO2 areas on the previous slide accounts for the higher energy expenditure. So we're also not seeing any differences in locomotor activity between the groups. Moving on, looking at tissue weights, we see that in both fiber groups, the colon length is slightly increased or significantly increased, probably due to the interaction of the fiber diet and the colon. And we're seeing that liver weight is decreased. Interestingly, pancreas mass is larger in both of the fiber groups. We're also seeing that the adipose depots are bigger in the double knockout mice on fiber compared to their wild type counterparts, which matches the weight and fat mass data from the beginning. Interest in here again in the epididymal weight. Interestingly, the interscapular brown adipose tissue here is increased in the double knockout mice on fiber, which could account for some of the increased energy expenditure we saw previously. So overall, this study has shown that increasing the fiber diet offers protection to the wild type mice, and this is not seen in their matched double knockout counterparts. As for the mechanism, we do not see any significant differences in food intake. However, we did see that the energy expenditure of the double knockout mice on fiber is increased. So overall, we have confirmed the role of whole body short chain fatty acid signaling through FFA2 and FFA3 in the protection against Western diet. And we believe that this protection is offered through alterations in energy expenditure and thermogenesis. While we have confirmed this, it raises more questions, especially in terms of the mechanism. So to dissect out the mechanism behind this, we are going to confirm the expected role of the microbial composition in short chain fatty acid production is not playing a bigger role than we think. We will also conduct tissue analysis to understand the role of the adipose tissue along with incretin and insulin hormone signaling in the protection against metabolic dysfunction. We are also planning on repeating the previous 25-week-long study with a couple of changes. We want to add a chow control group to metabolically characterize the double knockout mice over the same period of time, but without a dietary challenge. We additionally plan on conducting baseline metabolic and food intake analyses, along with testing baseline insulin sensitivity and secretion to confirm our assumption that the baseline for the double knockout mice are equal to the wild type mice. So to go back to the overall understanding, we have kind of confirmed the role of the receptors in the protection against metabolic dysfunction. To go over to AIM-2 now, we want to look at the effect of fiber on the microbiome and see if this can impact metabolic dysfunction, potentially even independent of the receptors. So when we eat a high-fat, high-sugar Western diet, we are seeing reduced microbial diversity and an overall metabolic dysfunction. However, a diet high in fiber can increase metabolic or microbial diversity and protect overall from metabolic dysfunction. So we're trying to see where this effect comes from again. Is it the direct increase in microbial diversity in itself protecting the metabolic dysfunction? Or rather, is it another effect such as short chain fatty acid signaling? Or of course, both. So AIM-2 looks into more whether the protection of fiber against the Western diet comes from the heightened microbial diversity, in addition to the short chain fatty acid signaling we have just proved. So when we study the effect of a fiber supplement to Western diet, we're seeing that almost immediately after the diet starts here at week 1 or 2, the mouse weights start increasing steadily. However, the protection against fiber diet doesn't really emerge until 3 or 4 weeks after the diet starts. So we think this could be due to the effect of fiber on the microbiome starting to kick in a couple weeks into the diet, once the microbial diversity develops. Therefore, we're proposing an acute SNAP study exploring the role of the microbiome in fiber-mediated protection against the Western diet. So to do this, we will conduct a fecal microbiome transplant study. We're going to create a pseudo germ-free mouse via a 3-day antibiotic treatment. Mice will then be transplanted with human gut microbiomes from volunteers with either a high fiber diet or a low fiber diet. These microbiomes will be either highly diverse from the high fiber diet or not from the low fiber diet. We will then conduct a 2-week acute study to see the inherent effects of the highly diverse microbiota. Mice will be challenged with one of 4 diets, either a low-fat or Western diet, or the low-fat or Western diet supplemented with fiber. Metabolic analysis will be conducted for 5 days here, 1 week into the study, to allow for the microbial transplant to settle and some dietary phenotypes to develop. So again, high or low diversity microbiomes will be transplanted into a mouse and fed one of these 4 diets. In this, what we expect to see with the low-fat diet is honestly not much of a significant effect. Without dietary fiber in the highly diverse microbiome, we're not expecting much, just a control. Similarly, we don't expect an effect when we add fiber to the low-fat diet, as there is nothing really to protect it from in the absence of the obesogenic Western diet. When the mice are fed just a plain Western diet with no fiber supplementation, we expect that there will be little to no protective effect from the highly diverse microbiome without the additional dietary fiber supplementation. And lastly, when the gut microbiome preconditioned with a high fiber diet is fed a Western diet with fiber supplementation, we do expect it to protect against an acute Western diet challenge. Therefore, with this, we plan to see the highly diverse microbiome may have a minor protective effect against the Western diet without available dietary fiber. However, if the fiber is available in the diet, the diverse microbiome will protect against an acute Western diet, presumably through the short-chain fatty acid production and FFA2 and FFA3 signaling. We haven't conducted this study yet, but we plan on doing it in the near future. I just wanted to take this moment to thank everyone in the Layden Lab for all their help. It's been a wonderful first year. And thank you everyone here for allowing me the chance to speak today, and I'll take any questions.